Hello everyone, I'm first presenter Ho Young Lee and I will present about classification and cluster analysis using G/G Tofms. Why I choose this paper, the research of G/G Tofms, on the research of G/G Tofms, the classification and cluster analysis were widely used for data processing, so I briefly suggest of these techniques. First is introduction. So on G/G Tofms, non-target analysis was conducted and thousands of peaks were identified. So that is the most big problem, that is the data processing was defined by user selection of interesting compound, yes or not. So before, the classification or other statistical analysis were used for reliability criteria based on detected peak for our interesting compound using many algorithms or statistical analysis. So before data analysis, as you know, that we got the sampling and had pre-treatment and instrumental analysis using G/G Tofms. And we got a larger number of chromatograph features and peaks. And these analysis results come through the many process of data processing, from chromatograph feature, peak identification, classification, and categorization. Firstly, based on the analysis result, we got the peak information such as retention time and SN ratio. Based on this information, we got the peaks from this sample and analysis results. So generally, on the analysis result of G/G Tofms, thousands of peaks were identified on peak table. So it is an important point on this peak. The interesting compound were selected by our purpose. So based on this program and the other statistical analysis was focused on the classification. So the classification is not widely used word, but I understand this classification is same with the grouping of compound which was our interesting compound. So based on basic program of this instrument, there are many many functions were suggested on the basic program. So I firstly suggest the basic program function and other further statistical analysis. So from this basic data processing function, so after that we can conduct the categorization for further studies such as cluster analysis or other PCA. So on this presentation, I just focused on the cluster analysis. So this is the first window of Chromatoph software. There is a basic software for G/G Tofms. So the left side is a tree of this software. So the first part is for the analytical method and analysis results. So we can write method file and check the analysis results. And after that, we can conduct the library search and quantification on this function. And finally, we can conduct the classification and filtration of our interesting compound using this function and trees. So I present about these two functions, statistical compare and classification. So before this classification, we needed to conduct the chromatographic feature and peak identification. So before mentioned, on this chromatogram, there are many peaks of their peak width and SN ratio. So using peak width and SN ratio or match require percentage. So we filter which peak is compound and which peak is not compound. So is a result of peak table. So on this chromatogram, there are black dot is a peak marker based on this finding program. So after peak identification, we can conduct two types of classification. The first is a classification function, and second is a statistical compare function. So there's some confusing of wording there because my presentation title is classification. So that classification means grouping of compounds, and this classification is just function of name. So first is classification is basically this function focused on the selection by groups characteristic. So based on identified peaks, that is number of these group, maybe thousands or hundreds group. Based on this, we filtered our interesting group on 1 or 2 or 3. For example, our interesting group is POPs, so we can get the chlorine or brominated compound based on this classification. And otherwise, the statistical compare is compare between the samples. So we analyze many number of samples, and we can divide the sample based on the sample characteristics such as industrial or rural area. And we got the commonly detected compound on the same sampling group. So on this number of sample, we divided on the group 1 and group 2. Maybe, for example, there is a rural and industrial area. And if the 3 sample in industrial area, and there are 100 commonly detected compounds, otherwise 4 sample in rural industrial area, there is a 200 compound can be detected. So in this presentation, I focused on the statistical compare function. So before statistical compare function, I briefly explained the classification function. So on the chromatogram, there is first retention time and second retention time, and the eluted and reserved peak shows the similar property if there is located similarly. So we got manually draw the rectangular or circular, so we can define this group is carbon number is 11 and 12 and 13 and 14. So these classified these circle were applied to other samples. And also before mentioned that is the other function is script function, that is a basic code software using VB script. So on this function that based on the mass spectrometer, we can identify that compound contain bromine or nitro or chlorine compounds. So based on this classification, we can identify the similar property compound on just one sample. So on this function, there is an advantage of application of chromatographic and mass spectra information for criteria of filtering. And next is statistical compare function. So before mentioned, we analyze many number of samples and divided the group by our user defined, and based on one group, and we conducted on 3 functions: analytics, oligomers, basic statistics, and Fisher ratio. So firstly is analytics oligomers. So we can divide it on group 1 and group 2 based on our sample properties, such as high concentration or low concentration. So based on this group, we can identify all detected compounds. So for example, on group 1, there is a 3 sample. So 300, 400, or 500 were detected each sample respectively, but we can identify 100 sample or coil detected on this sample. So this concept is most important because this non-target analysis shows have higher reliability with this duplicate analysis. So, and after that, we can compare the group 1 and group 2 filtered compound using this retention time and mass spectra information. And after then, we got the co-detected compound, and on the software, they are the basic statistics such as average or standard deviation were automatically calculated. And finally, the Fisher ratio also can be calculated. So I prepare some example of statistical compare. So click this statistics compare tree and make a new pile. And there are some 3 tables. So first is class table and sample table and compound table. So firstly, we can define the class table such as high pollution, episode 1, episode 2, and clean day. And each class we can add sample to class. For example, in the high pollution episode, we can input 2 sample and 3 sample and 5 sample respectively. So we can check the sample by each class. And after that, we can show the detailed information each samples and samples class, and we got the scale of these samples. And that we conducted analysis, and the co-eluted compounds were identified on this table with their area or other quantified mass information. And also on this statistical compare, they suggest the other optional, there are other options based such as normalization or export to Excel. So firstly, if we select the three compounds, they show some detailed information of each samples. And we can normalize by internal standards on the software. And after normalization, we can export all compounds for Excel to further statistical analysis. So many papers focused on the other statistical analysis based on these co-eluted compounds. So we can export this co-eluted information to Excel, and we can apply to other statistical analysis such as cluster analysis and PCA. So I just briefly present about cluster analysis for grouping of this information. So the cluster analysis is just the application of algorithm of that group of similar objects. So based on their algorithm, there is many types of cluster analysis such as K-mean or HCA. So I select this connectively model. HCA is the most understandable for easily. So this figure shows the concept of the hierarchical cluster analysis. So based on this sample, so sample was located based on their distance between sample, and which 2 samples which located nearest, they were co-eluted one cluster. So there is a step-by-step, the cluster was co-eluted, and finally one cluster was combined. So on the raw data, they was relocated based on their distance on matrix, and they was combined based on their dissimilarity. So for example, there is 7 sample on the data set, and this value means the dissimilarity. So the low value means there are 2 sample was show highest similarity. So sample B and sample F show the lowest value of dissimilarity. So these B and F were correlated as the same cluster. So finally, we got the dendrogram figure from this cluster analysis. And there are many options of the metrics and linkage criteria of the cluster analysis. So based on our data property and our research purpose, then we can select the detailed option. So for conducting cluster analysis, there are many software such as SPSS or MATLAB or Origin. So I will briefly conduct this cluster analysis using some type of software. So I just show some example of the software. The firstly is SPSS. So this data set means they are our preferred correlated compound on same one group. So the sample number is 16 and the compound number is 100, almost 100. And this all group were inputted using on cluster analysis and hierarchical cluster analysis, and the value were inputted the variable, and there are other options of the matrix option and linkage option, and there are other displays option of dendrogram or the other cluster information. So and we can get the cluster dendrogram of this cluster based on this cluster analysis, and based on the cluster cluster schedule information, we cut the number of clusters such as 4 or 7. So these cutting value were divided these coefficients. So basically this number of stages, the coefficients was increased, and the one step shows the highest increasing value. So on this some reference, there is a highest increasing value. So when the coefficient is highestly increased, so we need to select this cluster number. And also, on the MATLAB, then we can input the other compound. We can write some code for the cluster analysis, and many paper and reference suggest some coding for cluster analysis. So we can conduct it also using in MATLAB. And also in Origin Pro program, they suggest the application of the heat map with dendrogram. So we can download on the website for cluster analysis with heat map. And we can input the data set of our correlated compounds and input the other values such as cluster method or distance type. So we got the dendrogram of this x-axis and y-axis with the heat map. So in this figure, the y-axis means the samples, and x-axis means the compounds. So this grouped compound shows the similar property and similar characteristics. So if there are some compound on this group shows the important characteristic, and then we can need to interpret other compound on the same group. So finally, why I choose this paper? Firstly, mentioned the many papers suggest this data processing procedure. So on this book, they suggest the analysis results through the data processing, and finally data mining using cluster analysis. So on this paper, they conducted cluster analysis, and they select these two clusters is the interest. This is important group, and on this group they conducted the PCA using these two groups. And the other paper, they also conducted the heat map with dendrogram, and they suggest the cluster 3 shows the interesting compound because some compounds were well known in previous, and the other compound can be a new indicator or marker with similar properties. So I think if we got the large number of data set of the analysis, this data mining or this data processing from identified peak, and there are some criteria for filtering, and finally we can conduct the cluster analysis or something other statistical analysis. So maybe I study more about these statistical tools, and after that I want to more detailed interpretation of this analysis. Thank you for the listening.